Hey, welcome to Planetterrian Daily, episode 60. Today is May 11th, 2026. Let's get into today's research and health developments. Over 1,000 genetic switches function differently in female immune cells, helping explain women's higher rates of autoimmune disease. Researchers mapped regulatory elements active in immune cells and found more than 1,000 that operate differently between women and men. The differences align with the well-documented higher incidence of autoimmune conditions in women and point to sex-specific immune regulation as a core biological feature rather than a secondary effect. This large-scale mapping reveals that immune regulation is not a uniform process across sexes, but is instead tuned by thousands of distinct molecular controls. Understanding these switches could guide more precise approaches to treating or preventing conditions that disproportionately affect women. The work shows how these elements shape daily immune responses at the most basic level of gene control. Tracking them over time may help refine prevention strategies tailored to individual biology rather than broad population averages. You've probably heard that the immune system operates the same way in everyone, with only minor variations from person to person. In reality, more than 1,000 genetic switches already behave differently in female immune cells compared with male ones. Right now, as you sit listening, those switches are shaping how your body recognizes and responds to threats in ways that differ by sex. One memorable figure is the count itself. Over 1,000 distinct regulatory elements show sex bias activity, a scale large enough to influence disease risk across entire populations. The next time you hear about autoimmune conditions or vaccine responses, remember that these differences begin at the level of gene control rather than emerging only after symptoms appear. Tracking how these switches respond to nutrition, sleep, or environmental exposures offers a concrete way to watch for new findings that could refine prevention strategies for both women and men. Now, shifting to a very different area of research, new analysis shows that thymic health continues to shape immune competence well beyond childhood. Reduced thymic output in adults correlates with weaker responses to new infections and vaccines. The findings emphasize that the thymus maintains immune diversity well beyond early life rather than shutting down after childhood. Results underscore the organ's ongoing role in overall immune resilience and its direct link to how effectively the body handles novel pathogens later in life. This ongoing output helps preserve a broad repertoire of immune cells that can recognize and respond to threats encountered for the first time in adulthood. The data suggests that preserving thymic function could become a larger focus in strategies aimed at supporting long-term immune health. On a different note, researchers at the Catholic University of Valencia are exploring collagen extracted from jellyfish caught as fishing bycatch. The material shows promise for cosmetics and biomedical applications, turning an underused marine resource into a lower-impact alternative to traditional collagen sources. By recovering this protein from animals already incidentally captured during routine fishing operations, the approach reduces waste while providing a scalable feedstock. Tests indicate the extracted collagen maintains structural properties suitable for tissue engineering and skin applications. This method avoids the environmental footprint associated with conventional animal-derived sources that require dedicated farming or harvesting. The work demonstrates how incidental marine resources can be redirected into practical biomedical materials without additional ecological strain. Having examined core immune mechanisms, shift now to a targeted genetic approach that addresses protein buildup inside the body without direct brain delivery. Delivery of a protective gene to the liver gene reduced amyloid plaques in the brains of Alzheimer's model mice. The therapy did not need to cross the blood-brain barrier, suggesting a peripheral route to central nervous system effects. This result points to new possibilities for treating protein accumulation diseases through distant organ targeting rather than invasive central interventions. By acting at the liver, the approach leverages natural clearance pathways that can influence brain pathology indirectly. The mouse data show measurable plaque reduction without requiring the therapeutic vector to enter neural tissue directly. Such findings open avenues for therapies that sidestep the challenges of blood-brain barrier penetration altogether. Leaving biological therapies behind, engineers at Harvard SAIS demonstrated the first interaction between a single quantum of vibrational energy and a single atomic spin. The result establishes a foundation for using sound waves rather than light or electricity to carry quantum information in future devices. This single phonon coupling marks a step toward mechanical systems that could integrate with existing quantum platforms. The demonstration shows that vibrational quanta can interact coherently with atomic spins under controlled laboratory conditions. Future devices might harness these mechanical vibrations for more compact or energy-efficient quantum processing elements. The work lays groundwork for exploring sound as a distinct carrier alongside photons and electrons in quantum architectures. Before we go, keep an eye on how peripheral gene therapies and sex-specific immune mapping might converge in upcoming studies of neurodegenerative and autoimmune overlap. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.